Welcome to this explainer. Today, we are jumping straight into what I like to call a theory to practice masterclass. We're going to build a rock-solid, textbook foundation on the absolute marvel that is human immunity. But we aren't just stopping at the theory. No way. We're going to immediately take that knowledge and use it to confidently crush a really complex trial exam question. Specifically, question 10 from the 2023 SBP Biology Paper 2. So get ready to see exactly how mastering these core concepts translates directly into maximizing your marks. Okay, let's dive into our game plan. We'll be covering the 3 lines of defense, the actions of antibodies, and the types of acquired immunity. Before taking all of that and applying it to our exam prep for both the inflammatory response and vaccines versus antiserums.
Section 1. The 3 lines of defense. Fortress human body. To truly wrap your head around immunity, I want you to picture the human body as a heavily guarded medieval fortress. Every single day, microscopic invaders are trying to breach your walls. But your body has this brilliantly layered security system designed to keep them out. Or, absolutely destroy them if they manage to get inside. Before we meet the guards, let's lock in our textbook terminology. Immunity is officially defined as the body's ability to fight infections caused by pathogens or other foreign objects through specific attacks. Now, what exactly is a pathogen? They're simply the microorganisms, like bacteria and viruses, that actually cause the disease. And on their surface, they carry antigens, which are basically the foreign chemical flags that trigger our immune defenses in the first place.
So what makes up the outer walls of our fortress? Well, that's the first line of defense. This is a non-specific barrier made of both physical and chemical shields. Physically, we have our tough outer layer of skin, but it's actually a chemical shield too. Your sweat and sebum contain lysozyme, which is an antimicrobial enzyme that literally dissolves bacterial cell walls. You'll also find lysozyme in your tears, saliva, and nasal mucus. Moving further down, we've got the mucous membranes trapping invaders in the respiratory tract and hydrochloric acid in the stomach just melting any bacteria hiding in your food. And if your skin gets a cut, the blood clotting mechanism immediately kicks in to seal up the breach.
But if those outer walls fall and a pathogen sneaks inside, the internal guards take over. This is the second line of defense, which is also nonspecific. It relies on 3 main responses. First, you get a fever, which turns up your body's heat to increase phagocytic activity. Second is phagocytosis itself, where white blood cells called phagocytes act like Pac-Man and literally engulf and digest the invading pathogens. And third is inflammation, which is that immediate swelling and redness you get in response to tissue damage. Actually, I want you to mentally highlight the word inflammation right now, because we're going to need that for our exam practice a bit later. Now, if the invaders somehow survive the first and second lines, the fortress deploys its elite special forces, the third line of defense. Unlike the first 2, this is a highly specific immune response. It relies on lymphocytes. These are specialized white blood cells that recognize the exact antigen of the invader and produce highly specific antibodies to destroy it. And crucially, they also create memory cells. These cells basically store the mugshot of that specific pathogen, so if it ever tries to invade again, the counterattack is massive and immediate. Section 2. Actions of antibodies. The 5 mechanisms. We know lymphocytes produce antibodies. But what do these elite proteins actually do once they're released into the bloodstream?
Well, there are 5 specific ways antibodies neutralize threats, and you absolutely need to know these terms. First up is neutralization. This is where antibodies bind directly with bacterial toxins to neutralize them. Second is agglutination. They basically clump or coagulate the pathogens together, which makes them a massive, easy target for phagocytes to gobble up. Third is precipitation. They react with dissolved antigens to form an insoluble complex that precipitates out, again, easily caught by phagocytes. Fourth is opsonization. Antibodies act as bright chemical markers, essentially painting the target so phagocytes can easily recognize them. And fifth is lysis. Antibodies combine with antigens to literally break down and burst the bacteria apart.
Section 3. Types of acquired immunity, active versus passive. So that covers our fortress defenses. But how do our bodies actually acquire these elite antibodies in the first place? It boils down to 2 overarching categories. The difference here is all about who is doing the heavy lifting. In active immunity, your own body's lymphocytes are producing their own antibodies in response to an antigen. Because your body did the work and created those memory cells, this protection remains for a long period of time. Passive immunity, on the flip side, means your body is just receiving ready-made antibodies from an external source. Because you didn't make them yourself, you don't get memory cells. The protection is immediate, which is fantastic in a life or death emergency, but it is strictly temporary.
So the crucial point is how we map these concepts to real-world scenarios. Both active and passive immunity can be acquired naturally or artificially. Natural active immunity is when you catch a disease, fight it off naturally, and recover. Artificial active is when you get a vaccine. In both of those cases, your body makes the antibodies. Natural passive immunity is a baby receiving antibodies through breastfeeding or across the placenta. Artificial passive is receiving an anti-serum injection, like antivenom for a snakebite. Lock this grid into your memory because it perfectly sets up our upcoming exam challenge. Section 4. Exam Prep Inflammatory Response, tackling question 10A. All right, theory time is officially over. Let's get into the practical masterclass.
Let's explicitly call back to that second line of defense we just learned about. The absolute key to getting full marks here is laying out the strict chronological sequence. When you get a cut, step 1, the damaged tissue releases a chemical called histamine. Step 2, this histamine causes the local blood capillaries to expand, bringing way more blood to the area. That's why it gets red and hot. Step 3, histamine also increases the permeability of those capillaries, essentially making them leaky. And finally, step 4, because they are leaky, phagocytes and clotting factors can escape the blood, accumulate in the infected area, and carry out phagocytosis to destroy the bacteria.
Look at how beautifully our logical steps map onto the exact marking scheme. Damaged tissue releasing histamine gets you point P1. Stating that histamine stimulates the response is P2. Histamine causing capillary expansion is P3. Allowing more blood flow is P4. Increasing permeability is P5, which leads to the accumulation of phagocytes for P6. Triggering blood clotting is P7, and the final act of phagocytosis is P8. Nailing any 6 of these gives you maximum marks. Using strict textbook terminology directly equals points. Section 5. Exam Prep, Vaccine versus Anti-Serum, tackling question 10B. It's time for the final boss battle of this explainer. This is a classic 10-mark essay question, asking you to compare the 2 methods of artificial immunity. And remember, to ace a compare question, you always need both similarities and differences.
We'll start with the similarities. Based on the marking scheme, to grab the first easy set of points, you just need to state the fundamental biological facts. Both vaccines and antiserums protect the body from infectious diseases. Both of them inherently involve antibodies and antigens in their mechanisms. And, tying right back to our grid from earlier, both are categorized as forms of artificial immunity.
And this brilliantly illustrates how to structure your differences for the remaining marks. You literally just contrast them directly. For type, a vaccine is artificial active immunity, while an antiserum is artificial passive immunity. For substance, a vaccine is a suspension of weakened or dead pathogens, but an antiserum is a serum already containing specific antibodies. For purpose, vaccines are given before infection for prevention, whereas antiserums are given for treatment when immediate protection is required right now. And for duration, vaccine immunity lasts for a long period of time, while antiserum immunity is strictly temporary and does not persist.
Now, looking directly at the actual marking rubric, check out points P4 through P11. Every single point we just mapped out in our table is exactly what the examiners are checking off. Having a solid grasp of the Chapter 11 textbook concepts, like knowing the difference between active and passive, or knowing what an antibody actually is, pretty much guarantees exam success when you structure it clearly. You even get extra points for mentioning booster doses for vaccines.
We've covered a whole lot of ground today, right from the first physical barriers of your skin all the way down to complex artificial immunities. The human body is just a miraculous, heavily defended fortress, with this incredibly adaptable biological machinery working inside you every single second of the day. So as we wrap up, I want to leave you with this provocative thought. Considering how efficiently your lymphocytes create memory cells to protect you today, how will your immune system adapt to the unseen, unknown viruses of tomorrow? Thank you so much for joining me for this explainer. Keep learning and keep appreciating the incredible science happening right inside your own body.